You would think lifting a small woman would be easy, but really it takes a series of muscles and processes to make this movement possible. Let's break it down to what happens in one muscle. A single skeletal muscle, such as the bicep muscle, consists of fascicles. Fascicles are made up of myofibers, which are encased by a cell membrane, the sarcolemma. The myofibers are filled with myofibrils, which consist of microfilaments.
This is a sarcomere, the basic functional unit of striated muscle. It's a highly ordered unit of thick and thin filament and is where the actual muscle contraction happens. The physical characteristics of a sarcomere include the A band, which is myosin, or a thick filament, an I band, which is the space between the Z disc and the edge of the myosin, the H band, which has only thick filament throughout it, and the Z line, which is what the thin filament attached to create the structure of a sarcomere.
Inside the microfilaments are thick and thin filaments. Thick filaments are characterized by the red Twizzler and are made of myosin. Myosin can be present at high energy or low energy state. The top filament represents high energy state, where the low structure is low energy state.
Myofibrils are built from 3 different kinds of proteins. Actin is a thin filament, which is represented by the yellow Play-Doh. Actin also has a myosin binding site, where myosin head binds during muscle contraction. 2 other proteins are also part of the thin filament, troponin and tropomyosin. Troponin is represented with the purple clay, and tropomyosin is represented by the black Twizzler. Tropomyosin is covering the myosin binding site on actin molecule during muscle relaxation. This prevents myosin from bonding to actin. Tropomyosin is held in place by troponin. When calcium is released from the sarcoplastic reticulum, it changes shape and binds to the troponin and pulls the tropomyosin away from the binding site on the actin. Troponin and tropomyosin function to regulate muscle contraction by covering and uncovering the muscle site on the actin. Calcium is also a key player.
As the action potential travels down the axon of the nerve, it arrives at the axon terminal, causing calcium channels in the terminal to open. This excessive influx of calcium pushes vesicles containing acetylcholine toward the end of the synaptic knob, shown by these green jelly beans here. Here the vesicles merge with the lipid bilayer of the knob. When the vesicles reach the synaptic knob, they release acetylcholine into the synaptic cleft, shown here by the green nerds. These nerds bond to the acetylcholine receptors, which are located on sodium channels. The sodium channels then open, letting in the pink sodium ions. The influx of positively charged sodium ions depolarizes the cell, creating a higher internal charge.
The function of the T tubules is to conduct a chemical signal from the surface of the cell. The terminal cisternae, the noodles, have calsequestrin, the purple netting, that store calcium, the yellow nerds. When the action potential arrives, the calcium is released. Thick myofilaments are found in the middle of the sarcomere. As long as calcium is present, the myosin head lets go of the ATP, the orange nerd, then flips the muscle and shortens. The myosin head binds to actin. The ATP, orange jelly bean, binds with the myosin head and pulls it off the active site.
After the action potential propagates down the sarcolemma, acetylcholinesterase, shown here by the green Play-Doh, bonds to the acetylcholine and breaks it out, thereby closing the sodium channels